It is my pleasure to introduce the next session of the symposium. In this session, we'll hear from Dr. Andrew Bate on how signal detection methods are evolving in the era of big data. These are tools that certainly have application when identifying possible safety concerns of new therapies. Dr. Bate is Vice President and Head of Safety Innovation and Analytics at GlaxoSmithKline. At GSK, he's a member of the Global Safety Leadership Team and is spearheading the development of next-generation safety capabilities. Previously, Dr. Bate was in the Global Epidemiology Leadership Team at Pfizer, where he oversaw the provision of epidemiologic analytic expertise for their worldwide safety activities. Prior to joining Pfizer, Dr. Bate was at the Uppsala Monitoring Center for more than 12 years. Dr. Bate has an extensive research record focused on development of methods and tools for the analysis of diverse types of real-world data. This includes spontaneous reporting systems, electronic health records, and insurance claims databases. He has more than 100 publications in peer-reviewed international journals on elements of safety surveillance, including the use of machine learning. It is exciting to welcome Dr. Bate to the symposium.
Hi, everyone. My name is Andrew Bate, and I'm Head of Safety Innovation and Analytics at GSK. It's my great pleasure to be with you today to talk about signal detection in the era of big data and be a part of this fantastic forum. A little bit about me before we get into the details of the presentation. As I said, I'm a full-time employee of GSK. Prior to that, I was at Pfizer in the epidemiology leadership team for 12 years. And prior to that, I was at the Uppsala Monitoring Center, the WHO Collaborating Center for International Drug Monitoring in Sweden for 12 years, where I headed up the research function. I'm also an adjunct associate professor of clinical pharmacology at NYU and of epidemiology at the London School of Hygiene and Tropical Medicine. So today I wanted to talk about signal detection and big data, because the world of data has changed massively since the dawn of pharmacovigilance, which was in terms of systematic safety surveillance in the 60s. And now we have all these fantastic electronic forms of data that we can link together from clinical databases, lab test findings, registries, hospital discharge notes, biobanks, other administrative databases, our insurance claims databases, and increasingly the Internet of Medical Things or Internet of Things, where we have the ability to link cardiograms, cardiology monitoring, data from our Fitbits and other applications. So we have this wonderful potential of very rich, very diverse data streams that have potential value for healthcare. And big data in pharmacovigilance, the opportunity is we can store data like never before. We can analyze using more and more sophisticated methods. Computationally expensive approaches that weren't feasible many years ago are now tractable, and more sophisticated visualization and reporting. So there's a huge amount of potential in terms of the data that's out there that we can use for pharmacovigilance. And yet, at its core, much of pharmacovigilance, certainly in terms of signal detection, is focused on spontaneous reports still. Before I go further, though, I think it's important that we're aligned, at least for this presentation, on what I mean by signals and therefore signal detection. And there are multiple different definitions still used, but I'm using the CIOMS 8 definition of a signal of suspected causality, which is information that arises from one or multiple sources, including observations and experiments, which suggests a new potentially causal association or new aspect of a known association between an intervention and an event or set of related events, either adverse or beneficial, that is judged to be of sufficient likelihood to justify verificatory action. Now, the very fact that CIOMS felt the need to come up with a new definition just over a decade ago, I think, is in part very much driven by the way that pharmacovigilance has become more and more about leveraging and using a wide range of data sources holistically as we think about all of the aspects of pharmacovigilance. And you'll see in this first part of the definition, from one or multiple sources, signal detection is no longer seen as just about one data source. Observations and experiments, so how do we use real world data as well as spontaneous reports and experiments, as well as a new potentially and suggesting a new potentially causal association. So there's something about it that means that we need to investigate it further, but it isn't causal yet. It's a question mark that requires further analysis. And it has sufficient likelihood. There's something about it that makes you think it needs to be investigated further. It justifies verificatory action, where it might be closed out and decided that it isn't something that warrants further analysis, or it may be that it needs further activities. And as I say, still in terms of signal detection, spontaneous reporting is the cornerstone of adverse drug reaction and adverse events following vaccination as an approach. So what is it that's so good about spontaneous reports? Well, they're suspected adverse drug reactions of previously unknown drug adverse reactions. So there's no need to be definitive. It's suspicions. So it's an opportunity to collect data where there's some concern going on. The challenge, though, is that they're reports of adverse events, something that's happened after a therapeutic intervention of some sort. They aren't necessarily true adverse drug reactions or true adverse reactions to vaccines. Often they may be temporary, or they will be temporarily associated, because that will be what triggered the suspicion often, right? But not caused by the drug. So vaccination occurred, adverse event, fell over, occurred. That's temporarily associated. There's no reason to believe that the vaccine caused the event. Many things occur for many reasons. But if things occur newly after a therapeutic intervention or immunization, then there may be some suspicion that the product is to do with it, which can trigger an adverse event report into databases. And we use this sort of data for hypothesis generation of possible side effects, and this is signal detection. As I think I've already alluded to there are many major limitations for spontaneous reporting, and it's critical that we as a field are very cognizant of these limitations and consider them in the inference and the analysis of how we think about the data. Well, there are many limitations and many papers that discuss this in detail, but just to summarize some of the key ones: limited clinical information and reports often, and satisfactory secondary case evaluation is not always possible. Very frustrating when there are some indications of data that's useful, but not really enough to really assess fully the case. Then we have to accept that not all ADRs that occur will be recognized as drug induced by a healthcare professional or consumer and reported. So some will be missed. And some of those that, even if they are suspected, may not be reported. And there's a very famous paper back in the 60s and early 70s from Bill Inman on the 7 deadly sins, the reasons why reports don't come in. But we always have to be very conscious of the opposite, that suspicion can mistakenly rest on the drug, and that coincidental spontaneous reports will result. We don't have control information as part of routinely collected spontaneously reported systems. We don't know what would have happened to the patient if they hadn't had the medicine. And we don't know the total utilization of the drug, certainly in these international systems. And we don't know the number of people who've had the drug and haven't had an adverse event. So in the epidemiologic 2x2 table of drug, taken the drug, not taken the drug, adverse event occurred, didn't occur, we don't know the B, C, and D boxes, and we only have a limited, we have a kind of an estimate of the A box that may be an overestimate or an underestimate. So there are fundamental limitations for spontaneous reports, which is why it's a tool for signal detection and can't be used for incidence estimation. So it's a tool for hypothesis generation, and we use other data streams to further investigate. So why then do we rely so much on spontaneous reports? Well, despite these limitations, it's a fantastically sensitive system in that we have a systematic approach for collecting suspicions of adverse events globally and in a timely manner, reporting them into us as pharmaceutical companies, directly to healthcare regulators, and to sharing in a systematic way across the whole system. And we're seeing more and more reports. This is an old slide that from 2005, showing exponential increase in the WHO database of adverse events collected from around the world, which at that point was over 3.5 million. And I deliberately keep this old slide because now it's at, it was over 10 million by 2015, and I believe it's just gone over 28 million in 2021, September 2021. So the number of reports in these repositories is massively increasing. I do think, though, it's critically important that we remember that there are emerging potential signals in this data, but there's also many of these reports that are indicative of concerns about medicinal products, which is important to think about, but are unlikely to represent new emerging information that will affect benefit risk. So well-established products that have been used for 50 years with adverse events that are labeled where we understand the mechanism, new adverse event reports are very sad and represent consumer or clinical concerns, but they are not likely to impact our benefit risk profile. And we need to think about that in our pharmacovigilance systems and how we analyze these data sources. The sheer volume of data increase has led to the opportunity and the necessity to use quantitative signal detection type approaches as part of these data sources. And this is a very old slide that I still like to show because I think the schematic of how we think about signal management is fundamentally quite similar now to how it was 20 years ago. So if you look at this slide and this circular flow, on the far left you have drug safety data coming in. Then we tag every single drug, vaccine, event pair in our databases with some sort of a metric of some sort, which I'm going to call a measure of disproportionality and come back to in a moment. We use these metrics to somehow then triage the data, quantitatively or qualitatively, to find disproportional recording. This is not a signal of suspected causality, but to somehow see what quantitatively stands out in the data source. And then we focus our clinical review on those reports and those pairs and clinical evaluation. And some of those go on to become signals of suspected causality. And then further on, and it's an ongoing process, hence the circular nature of it. Now, the reason I say this slide is that the role of quantitative metrics is to focus the attention on the clinical review, not bypass it. And this is in addition to other follow-up tools, direct clinical review of the case reports as they come in, and other triaging systems. But the role of quantitative signal detection is to focus the attention on unexpected clusters of cases, etc., that are more likely to represent emerging safety issues. And that type of approach is very much used today. Here's an example of a signal detected prospectively using this sort of approach in the Vigibase data. And most of these, in fact, all of these examples are published, and I'm very happy to share references afterwards if you'd like to read more about it. But here you see a measure of disproportionality, which in this case is the information component, is shown on the y-axis. And on the x-axis, we have time. And this is looking at how reporting into the WHO database, Vigibase, has come in over time. As we rolled back the database, we can see cumulatively how this metric, this information component, has changed. And I don't want to get into the details of measures of disproportionality because there's many presentations and publications that explain it in detail. But the core idea is that you look for unexpectedly frequent reporting of a particular pair relative to the generality of the database. And this metric is logarithmic. So 0 means I'm seeing about as many reports as I'd expect. And positive values mean we're seeing more reports. So for the anti-epileptic topiramate and glaucoma, what we see is over time, we start on the left with an information component of 0. And then as reports began to come in, the first report came in the second quarter of 1996. We see the information component suddenly shoot up because we're observing more than we'd expect, but wide confidence intervals because there's very little data. Then over time, the information component wanes. Why? Not because that report went away, but because we're seeing general reporting of topiramate and general reporting of glaucoma. And then suddenly, as you see around the 2000 time, the number of reports begins to increase suddenly, and the information component increases positively. We're showing 95% confidence intervals here. And you see then in the second quarter of 2000, this was quantitatively highlighted. Not that it was a signal of suspected causality. No, it was a signal of disproportional recording, which triggered clinical review. It then became a WHO signal, which was communicated out to regulatory authorities, and subsequently it was a Dear Healthcare Professional letter. Now, I'm not saying the WHO signal was causally related or led to the letter, but it was at least temporally related in the sense that this was highlighted and under review in the WHO system before it became a known issue. And there are many other examples of where quantitative signal detection has triggered the review that's led to a full signal. So spontaneous reporting systems like this are now widespread use across the whole system. So one of the things I'd like to ask and question, though, is that while, yes, we can leverage and make the most of these large data sets, we're spending an awful lot of time, if you remember, I've talked about big data at the beginning, around our processes for how we handle ICSRs, very manual systems, very complex systems, and this applies to pharmaceutical industry and regulators, because we have reports coming in, data ingestion occurring from all these different sources, call centers, agencies, partners, across companies, literature, etc., etc. And we're putting them and databasing them into our separate databases, and then sharing these reports with other companies, regulators, and putting them into supra-country national databases and other databases. So the same reports exist in different instances and variations all over the place in different places. And we have extremely complex, very intricate systems for audits, etc., existing across all of these separate systems. So a question then is, are we now in this era of big data, is it time to start thinking about our spontaneous reporting systems and how we can do things more effectively and different, and how do we begin to think about leveraging these other data sources? So as you can perhaps imagine, we have very complex, intricate systems, and Transcelerate, which is a cross-industry collaboration, has published on this idea, and I'm involved with Transcelerate, I'm happy to say, because I very much enjoy the work they do. An overall generic process of how a case report is looked at. And it seems quite simple, right? We take adverse event reports, we intake them, we process them, we report them to health authorities and business parties, and then do signal detection and surveillance at the end of it. But actually, this is a really intricate process, and this is just a generic system. If we think about case intake, that's made up of many steps: acquisition, validation, duplicate detection, prioritization, translation, etc., etc. And it's not just intake. Processing is made up of multiple steps, some similar or slight variations of those we saw before. And the same applies to case reporting. So one of the things that's the exciting development in the context of big data is leveraging intelligent automation, whether that be rules-based systems or increasingly machine learning, to more effectively look at this whole pipeline, this data pipeline, this process system. And in the context of big data, one of my take homes really from today is that it won't just be around this data entry perspective. We need to think holistically about signal detection across the whole data stream and the leverage of these machine learning type techniques, not just in getting data in, but thinking about how we do the signal detection and analysis and bringing that thinking into our data ingestion pipelines. Just to give you a flavor, and obviously in the context of time, I can only give you a flavor of some of the types of innovation that are going on, but I want to give you a few examples. So for example, in terms of robotic process automation, we use it for data quality review. So automatically cases are located for processing, logged in the safety database, search for each provided case ID, open each case, extract the relevant data, update the output report. Open source XML extracts the relevant fields and updates the output reports, highlight the output reports, look for discrepancies, and then update the quality reviewed, upload it into the original cases into the safety database. Human activity at the beginning and the back end, and obviously flagging for issues that need to be manually reviewed. But we're seeing, and this is across industry and regulators, we're seeing more and more robotic process automation, rules-based systems for standardizing systematically, more effectively handling data where we're doing the same things over and over again. And this is saving a huge amounts of time, which allows us to handle these huge, massive tsunamis of data that are now pouring into these systems. More sophisticated, here's an example, and again, I'm not going to get into the detail. You can read more about it if you're interested, and I can share details. Using natural language processing to review, we have to scan the literature to look for reportable adverse events, a huge task. And we've introduced routinely natural language processing for automated prioritization of safety literature review. And what this is able to do based on the nature of the attributes of case reports, rank reports. So rather than randomly working our way through all of the literature reports, we're able to show that using this sorts of approach, we were able to find the things that are most likely to include adverse event data that would be reportable at the top of the list, so that we could then focus and be much more effective in our ability to manually review those reports and then take action accordingly. So again, seeing routine use of natural language processing in the systems. I talked about machine learning as being increasingly used also and needs to be thought about holistically from end to end, and that includes signal detection. It would be a mistake to think this is all new. We've been looking at machine learning, and there are interesting examples for many, many years. And this is something that we, when I was in the WHO, we did nearly 2005, so in the early 2000s, nearly 20 years ago, where we were doing unsupervised pattern recognition from incomplete inputs to look for clusters in the adverse event data. And again, I don't want to go through the details of it, but simplistically, if you imagine you have prototype data, so in this case, 8 different patterns of adverse event reports of clusters, syndromes, if you will, and instead, as you know, spontaneous reporting is made up from incomplete samples, and actually the reports that come in look more like this. The plan is to try and replicate the original prototypes, even if we only get bits of the reports coming in. And we did this and looked at the neuroleptic malignant syndrome for haloperidol, the antipsychotic, and we're able to cluster a bunch of, in those days, HUART codes, so MEDRA codes, that were unexpectedly frequently co-occurring, so strong weights in the neural net, for those of you who know about machine learning. And the blue squares represent those with very strong positive weights, and this triggered this grouping, this clustering of all these adverse event terms together. The clustering landed in a stable configuration based on the individual counts of these particular adverse events that were occurring across the reports, but also pairwise highly reported pairs of adverse events that occurred together. And again, it's published in full if you're interested in the detail, but the take home is we've been looking at machine learning for many, many years in our signal detection as well as our data entry. And a question I'll come back to in a moment is, why then are we not using even more machine learning routinely? And just to finish on this slide, just to say that the MEDRA codes or HUART codes that came up in this cluster were all symptomatic of neuroleptic malignant syndrome. So it was a kind of a test positive result for this method. Another way you can use machine learning, and again, a published example, is for duplicate detection. Now, actually, many companies have very detailed data on the reports that means that you can identify with so-called safe fields that likely duplicates are coming in. But this specifically was work we done at Vigibase, where we had no safe fields. So no fields that if they were filled in, you were sure this is a duplicate of something else. And we were interested in developing a machine learning algorithm to find unexpectedly similar reports that might be indicative of duplicates. And again, you can see an analogy here with quantitative signal detection. Our aim was not to say these are definite duplicates, but to find things that are more likely to be duplicates that for clinical review or further follow-up. And here the core concept was unexpectedly similar. So here you see two reports, patient age, gender, country, substance, ADR terms, onset date, outcome, all identical. Would anyone here say that they are likely, very confident that these are two duplicate reports? Absolutely not, because these are common fields. It's often the case, sadly, that onset date may be missing. Rash is the most commonly reported adverse event term, commonly reported drug, huge database of adverse events. And the US is the biggest reporter into Vigibase, the WHO database. So instead, what we developed was we trained a neural net, a clustering algorithm that weighed unexpectedly commonly different records as low weight, whereas things very similar, very rarely similar by chance, would weigh very strongly. And then you search for clusters of reports that are very similar, but you just would never guess that they were like the very rare adverse events, things that are unexpectedly highly similar. So informative similarity rather than just similar. And then you cluster the adverse event combinations together. And then you can start looking at each field separately and look at how strong the weighting is that these reports on this field are similar or not. And then you can basically add them together and get an overall score. So you're rewarding positive weights for things that are very similar and very unexpected that they could be so similar, and negative weights for things that are very different. Add it all together and you get an overall score. The score, if it's very high, these two reports are really informatively similar. Very low score, these are very, very different from one another. And you can see here on the right, the distribution in general of any random pair of case reports in the database, kind of normally distributed around 0. And then for known duplicates, you see much higher scores. So this was the test set that we looked at in the database. And then you can use this to basically generate ranked lists of likely duplicates. Again, you can read more about the details if you're interested, but here is a typical example of genuine machine learning where you train the network in terms of what is unexpectedly similar or not based on the generality of reporting in a database, train the weights, and then look at new emerging data to find emerging duplicates. So some general findings as we begin to talk in general about machine learning and what some of the barriers may be. Volume of training data is critical. If you don't have enough data, you can't generalize good models, and they won't train good models, and they won't generalize well. It was difficult for the duplicate detection in terms of you don't have a simple yes/no criteria. It's a ranking. How far do you go down the list? I think that's challenging for implementation in systems. The need for ongoing training of the algorithm as data evolves and skews perhaps over time, as well as people using the system. And then when you start clustering things, you don't always get what you'd expect. There may be other reasons where you're finding unexpectedly similar reports, and that would be something I'd be happy to discuss for another day. But we found in our research even some really interesting clusters of reports coming up in this work. Adverse event reports, spontaneous reports, are limited in the data they have in there. So linking to other types of data is something that adds value and enables even more, and will in the future, I think, enable even richer machine learning. And even this idea, I wouldn't want to claim as new. This is a paper from nearly 20 years ago. Sorry, more than 20 years ago, nearly 30 years ago, from the early 90s. And what they did was they did training of adverse event data based on known receptor activity of antidepressants. And their idea was to be able to start subclassifying based on the known receptor activity of the sorts of adverse that came in. Now, whether the results were good, whether it's generalizable, isn't my point. My point is that bringing in that richer extra data of receptor activity allows us to begin to do extra machine learning, and we're going to see more and more of that as we move forwards. Not going to go through this in detail. Happy to share slides with you all. Just to say all sorts of different types of opportunity for using machine learning, and we're going to see more and more of it as we move forwards, from optical character recognition to supervised pattern recognition to clustering, etc., etc. How then, in terms of intelligent automation using these systems, what do we need to think about? And we need to be holistic. We need to think about pharmacovigilance system, training, operations, processes, etc. We need to think about the information technology to enable widespread use of machine learning from end to end, and also how we do testing and validation, etc. So there's a lot. It's not just about having the science and the research, but all of the other issues that go into embedding in routine capability. And it does require a rethinking of how the paradigm of how we do pharmacovigilance. There are multiple barriers which are, in my opinion, are very much surmountable, and we're seeing them gradually beginning to come down for widespread adoption of ML-based automation, ensuring credibility and interpretability of outputs. We need to be focused on problems where we have enough rich training and test data, and if not, find ways to focus energy or get more data, gold standards, reference sets for showing it's working, and then really have appropriate and recognized levels of performance accepted not just by ourselves as we test algorithms, but by all stakeholders. Just as I come towards the end, though, I've talked about big data at the beginning. There's a huge focus on how we more effectively use spontaneous reports and focus on where spontaneous reports best. That requires us also thinking about where other data sources are best. And in terms of healthcare databases, real world data claims, insurance claims and electronic medical records, we've used these data sets at the far right for signal evaluation for years and years. We're increasingly, through Sentinel and other systems, using them for rapid query capability for signal refinement to get ideas about emerging issues across networks of database. And we're increasingly beginning to explore how these data sets can be used for signal detection and where they're particularly valuable compared to spontaneous reports. And I think that is very much the future, as we see where spontaneous reports focuses where it's best for things like agranulocytosis, Stevens-Johnson, and where other types of signals might emerge from other forms of data. So it's emerging in signal detection and routine capability. I mentioned Sentinel, and I know we're talking about this in the conference, so I'm not going to talk about it in detail, but here on the left, we have this Sentinel system, this fantastic system with routine capability to query in a distributed network data from many different data sources. And similarly, then we have these sorts of capabilities through purpose-built or vendor-specific tools specific for pharmacovigilance to run analyses across multiple different data sources to do similar sorts of work. And this has been one of the massive advances, I think, in the last 10 years that we see this routine capability for rapid evaluation in a big data context across lots of different data sources. Just to say, signal detection, how do you do it? Lots of different approaches being explored. One idea in EMRs is, and this is some work I was involved in, compare, in contrast to spontaneous reports, we have data before, we have control windows, as well as after. So can we look at unexpectedly different things that happen after an exposure than happened in the medical record many months or years before? One way we can look for systematic, automated look for unexpected changes in recording of things in medical records compared to the time before. And here we show the idea of some sort of a value on the left there going along, and suddenly a normalized value suddenly changes at the time of exposure that might be indicative of an emerging signal. And it's very much a nascent field, I would still argue, and you're seeing lots of interesting methods work there, but certainly this is something that's going to advance. Again, whistle-stop tour here, just to highlight some of the issues. The ability to extract with natural language processing from the unstructured data on electronic medical records. For example, we did some work on acute liver disease, is also very rich for better signal detection and surveillance capability. And I know others are talking about that in this conference. And we showed that we could identify acute liver disease much earlier using unstructured data than with just the structured data alone in some work a few years ago. And then the last one, and then I will wrap up, is social media data. I think the mistake we make, there's been much research saying social media is limited in signal detection. I think it is for many things, but we need to remember that there's a whole range of different data sources that we put under this umbrella term, and that actually using social media in the right way for the right aspects of pharmacovigilance, I think is going to be increasingly important moving forwards, and make the most of it, strengths and limitations, and not try and replicate what we do with, say, spontaneous reports, and use it differently. IMI Web Radar have come up with some great findings for it, and really making that point that spontaneous reporting is a key thing, and that really broad statistical signal detection isn't recommended with social media based on their analyses. But one thing we did recently, think of the longitudinal richness of some of the social media insights, where wording, and I know you can't read this in detail, but I just want to make the point that some of the posts on social media are incredibly rich, and we did some work showing that longitudinally you can learn much more than you can with appropriate systems in place from individual posts, and I think that's a really rich source of data that we need to explore further. So let me wrap up. I'm conscious time's running here. In the 60s, we had spontaneous reports and clinical trial data and not much else, the odd primary data collection study going on. Now we have huge amounts of spontaneous reports, loads of real world data sources, all these different types of social media, not to mention things like the internet and medical things, all these different data sources. And we really need to rethink pharmacovigilance in this era of big data from the ground upwards. We shouldn't just try and do everything with spontaneous reporting. Maybe some of the reporting systems we have in place aren't needed as they are at the moment. Can we be more creative and think about how we use this whole wealth of rich data for advancing our signal detection capability and leveraging these emerging technologies? It's an incredibly exhilarating time to work in pharmacovigilance. There's huge amounts of exciting innovation possibilities going on, and I hope that me giving a high-level overview and a flavor of some of the things going on it is helpful, and has really shown that. It's driven by more data and more technologies. Spontaneous reporting isn't going to go away. It has a critical role, but it has a critical role, and some of the things that we've used spontaneous reporting in the past, I think, are better replaced with other data streams. And we need to think about how we really think about big data as we move forwards. Signal detection is only one part of a robust overall signal management system, and we need to really think about the further opportunities for advancing use of advanced analytics and other data sources and linkage across data sources. And safety surveillance is one essential component of an overall continual assessment of benefit risk. Thank you very much for your time.